Hello, everyone. Welcome to the 23rd installment of the PhenoTips Speaker Series. Today's episode is part of our Rare Disease Day awareness celebrations, as today, being the last day of February, is Rare Disease Day. And we're highlighting some of the incredible work being done by genetics professionals. My name is Dr. Paweł Buczkowicz. I'm the COO and VP of Scientific and Medical Affairs at PhenoTips and your host for today's webinar. I'll be moderating the audience Q&A discussion after today's presentation, which is on the topic of solving the unsolvable with Care4Rare. And our speaker is Dr. Kym Boycott, a clinical geneticist and senior scientist at the Children's Hospital of Eastern Ontario and professor of pediatrics at the University of Ottawa. Thank you, everyone, for joining us today. And before we jump into the presentation, I'll do a little bit of housekeeping. The presentation portion will be approximately 40 to 45 minutes long, with an audience Q&A in the last 10 or 15 minutes remaining. You can submit your questions through the Zoom's Q&A box at the bottom of your screen, and we'll take them at the end of the presentation. Since around 2020, PhenoTips has been sponsoring this speaker series, and we're excited to host this really special episode for Rare Disease Day. This is particularly important for us because PhenoTips was originally founded to advance rare disease research and diagnosis with a mission of improving outcomes for individuals living with rare diseases. PhenoTips' founding team originated at Sick Kids Hospital in Toronto, and the company itself arose from a collaborative research project with the collaboration between Sick Kids clinicians and researchers and the computer science department at the University of Toronto. Today, with our genomic health record software, we at PhenoTips are especially proud to support projects across the world to mainstream genomic healthcare initiatives and those that help advance diagnosis for the over 300 million individuals living with a rare disease. As global leaders in rare disease research and genomic medicine, CHIO has acted as a hub for national rare disease projects here in Canada, and that PhenoTips has been proud to be a part of, including Care4Rare, SolveRD, and by providing the Canadian rare disease research platform built on top of PhenoTips called Genomics4RD. So as I mentioned, today's speaker is Dr. Kym Boycott. Dr. Boycott is the professor of pediatrics at the University of Ottawa and the chair of the Department of Genetics at U of O. She is a clinical geneticist at the Children's Hospital of Eastern Ontario, as well as a senior scientist at the CHIO Research Institute. Dr. Boycott is a Tier 1 Canada Research Chair in Rare Disease Precision Health. Her research program bridges the clinical genomics to basic research aspect, focusing on understanding the molecular pathogenesis of rare diseases to improve the patient care and family well-being. She leads the National Care4Rare Canada Consortium, which integrates genomic and other omic technologies to improve the collective understanding of rare diseases, with a particular focus on solving the unsolved and the most difficult rare disease cases. To leverage some of those discoveries, she also co-leads the Canadian Rare Disease Models and Mechanisms Network, or RDMM network. This network was established to catalyze connections between newly discovered rare disease genes and basic scientists who can rapidly study them in model systems and model organisms. Globally, she moves the rare disease agenda forward as part of the Global Commission to End the Diagnostic Odyssey for Children. Dr. Boycott, I'm very much looking forward to your presentation, and the stage is yours.
Thanks, Paul. Okay. Okay, I'm going to share my screen. Give me 1 sec, and thank you for that kind introduction. There we go. Okay. You see that okay? Oops, sorry about that. Okay, thanks everyone. It's a pleasure to be here on Rare Disease Day to talk about solving the unsolvable. And that title links to our current research program that's part of the Care4Rare Canada Consortium called Care4Rare Solve, which is really aiming to enable a diagnosis for all rare genetic diseases. And this image that you can see on the title slide is a graphic that was drawn by Karina Keeley. So I think now probably 5 years ago, when we were talking about all the challenges that surround rare disease, and I think it's especially important to recognize that today. Today we're going to focus on diagnostic clarity, but I just want to recognize that there are many other challenges for families experiencing and living with a rare disease besides diagnostic clarity. And I'll talk a little bit about that towards the end. But as you can see from that image, it really links to natural history and connections with the world, and obviously something that's very important to everyone, which is treatment or appropriate management. So to start, rare genetic diseases, as was part of the introduction, affect about 300 million people worldwide. But what I'm going to focus on today is the subset of rare diseases that are monogenic. And that's a very important distinction because it's very confusing sometimes to see the different numbers that are used. When I'm talking about rare genetic diseases, I'm talking about those that affect 1 to 3% of the population and are caused by a single gene. So these are monogenic or Mendelian diseases. That's not to say that rare diseases as a whole don't have lots of genetic contributions. They most certainly do. And 80% of probably all rare diseases have genetic contributions. But when I speak about rare genetic diseases today, we're talking about those that have a monogenic contribution. And when we look at that category, there are probably about 6,000 rare genetic diseases now documented, caused by just over 4,000 genes, but there are likely thousands that remain unsolved, though that number I think probably is decreasing over time. And the challenge around diagnostic clarity really comes with this great image that many partners across the bottom put together a couple of years ago, which really is the embodiment of a diagnostic odyssey. And as you can see, as you follow that line along, that it's swirly, it loops back on itself. And this is really the patient experience with respect to diagnostic ambiguity. And what we're really focused on is trying to promote that into a straight line by ensuring that the right knowledge and technologies are available to individuals who have a suspected rare genetic disease at the beginning of their diagnostic journey. And so over the past 10 years, there's been a Canadian network called Care4Rare, which we started out as Forge, but that was a particular project. But it is, the consortium is called Care4Rare. And over the last 10 years, we've been working collaboratively across 21 sites in Canada to really think about discovering rare genetic diseases and enabling efficient diagnoses for families. And everything I'm going to say over the next, say, 40 minutes or so, I really need to recognize that this is the work of hundreds of clinicians, genetic counselors, scientists, informatics, individuals, etc., who have all come together to sort of help us collaboratively work on this set of challenges. So today I'm going to loop in 3 stories, and each of these stories really highlight the diagnostic odyssey. These are all patients that have been seen by me for more than 10 years. And so the first is Colton. I met him when he was 11 months old. Austin, I met when he was about 3. And then I'm going to talk at the very end about 11 families that I've been following since 2007 with a late onset cerebellar ataxia. And they all sort of really represent not only the diagnostic odyssey, but the fact that their diagnoses can only come out of research programs that use new technologies because their genomic mechanism is hiding from our usual toolbox that we have in the clinic. But I will come back to them as we go along. And so what we're focused on with Care4Rare is 2 things. So in order to enable a diagnosis for all rare genetic diseases, we have to do 2 things. One is facilitate access. So that's a timely diagnosis Second is we have to understand the mechanism of all the rare genetic diseases. And that obviously the mechanism feeds into the access and the knowledge once that has gone into the clinic. And we need to address both of those things. And we certainly have been focused on those from a Canadian perspective, which I'll tell you a little about a bit, and even more centrically from an Ontario perspective. So we all know that clinical genome-wide sequencing is a disruptive test. There's definitely no better test that we've ever had in genetics. And when I refer to clinical genome-wide sequencing, what I'm referring to is basically the focus on a broad interrogation of the coding 1% genome, 1% of the genome. And hundreds of thousands of rare disease patients have now been clinically sequenced worldwide. And the diagnostic yield for this test is usually in the 30%. And what it's doing is enabling this sort of image here on the left, which is the diagnostic odyssey experienced by one of my patients, Abby, and she's actually still undiagnosed. To be at least more efficiently streamlined for lots of individuals and their families to inform their care and enable their family well-being. Over the last, we've sort of known this has been a great test since 2013. And over the last 7-8 years in Canada, we've had variable access to this depending on where you live. And so we were quite interested in the Care4Rare program to facilitate translation of this new technology. And this is what I learned about health services research, which is probably very primitive, but you can either have a top-down approach, which is the great example of that is Genomics England, where there's new money coming in and an investment coming in to roll a new technology and let it roll down the hill. Not that that's not without its challenges, but you know what I mean. A lot of Europeans at laboratories rearrange their resources and shut down old tests and bring in new tests. And as long as they're budgeting their budget, it's okay. But where we live, we are kind of bottom up. Which really sometimes feels a little bit like rolling a rock up a hill. It's a very difficult approach. It's not an uncommon reality for many health jurisdictions. And so I found this a very therapeutic slide to draft several years ago, well, not several years, 2 years ago or so, that describes the process by which we moved forward, partly within Canada and then more specifically within my province of Ontario, to finally come out the other end in 2021. So it took us 8 years. To launch a program called Genome-Wide Sequencing Ontario as a pilot project over the past 2 years. It's now finished its 2-year pilot. To allow us to offer genome-wide sequencing in province for families with suspected rare genetic disease. And as you can look through that maze, you can see some of the things that we had to do around showing the diagnostic utility, developing Canadian best practice guidelines, defining on eligibility, thinking about data and consenting and the technical specifications of the test, building a program, implementing it, and then evaluating it. And you can see why bringing in new technologies into the healthcare system is such a challenge when there's no new net investment in that technology. And basically, you've got to make your case to have the resources for the test that will be a great addition for the diagnostic journey for these families. And so Genome-Wide Sequencing Ontario, which is a partnership between SickKids and CHIO, is finishing its 2 years this month, March. And basically what we've been able to do over the last 2 years is bring both exome and genome sequencing into the province and provide testing for eligible individuals. And we're over 3,000 individuals sequenced now. Our diagnostic yield is 31%. And most of the time we're able to return that result within 8 weeks. Whereas previously in the last few years prior to this, we could be waiting 5-6 months for these results when we sent this test for the few eligible patients out of country. And one of the key things in all this is really, once you'll get your diagnosis for about a third of your patients, but for those who are unsolved after that test, we have to really think about data sharing and a way to recontact families for research. And what's come out from the community is something supported by Genome Canada, which is called the All-for-One Precision Health Partnership. And what that really is focusing on is how do we take, de-silo some of this data that's being generated now within the clinical system and under the custodianship of the healthcare system, and freed it up for discovery with the appropriate consent of families. And so the way we're running things in Ontario here, you can just see this on our green box, which basically then is having families acknowledge their sharing of data, coded data between clinical reporting institutions to enable a better test. And the second is having a mechanism to consent families for recontact for research. And that's, in our experience, has been about 80% uptake with the recontact for research. And that gives families, no matter where you are, the opportunity to share their data for discovery, which I think is very important, and I'll show you why. So the vision for the All-for-One health data ecosystem is really a two-pronged solution. The first, which I was referring to, which is called All-for-One Clinical, and that is really sharing variants between clinical reporting laboratories such as SickKids and GEO, but also others in the country, to ensure quality assurance and quality improvement of the test. And this is really important given the fact that your ability to interpret this test is based on the number of controls you have. And so the more you have, the better. The second part is called All-for-One Connect, which is the opt-in registry to connect patients with research. And as I said, this will enable families to share their data no matter whether they're seen in Ottawa or Oshawa or Sudbury, for example. And so we've talked about, now we've got some reasonable access. Well, we could certainly debate that, but we have reasonable access in Ontario. It'll only improve with time. The foundation is there. Next, we need to focus on the mechanism, and that's really where our discovery and our research program spends a lot of its time. And over the past 10 years, we've enrolled over 3,000 families in this pipeline because they are undiagnosed after standard of care testing. And there are a lot of reasons they may be undiagnosed. And this is sort of a nice little picture to sort of explain that. And so the first reason that we'll focus on is the fact that the gene may not yet be discovered, or that disease gene association is not yet discovered. And we'll go through our approach to that. The other is that the genetic etiology may be undetectable to the technology that was performed. So can't find it on the exome, for example. And we'll all three of the stories I tell you today will be around that set of issues. And the last, which really then goes back to Rare Disease Day, is that the etiology may be more complex or not straightforward monogenic. And sharing of data will be even more important as we think about this set of diagnoses here, where there may be monogenic or environmental factors that also play a very important role, and how are we going to address those and discover those causes. So let's start though with the disease gene association may not yet be discovered. And many of you know that if you have, say, for example, clinical exome sequencing 3 years ago, and you have reanalysis of that existing data, say 2 to 3 years later, you're probably going to have an increase in your diagnostic yield by about 5 to 6%, which means that those families are now diagnosed because within that intervening year, we had almost 1,000 disease gene association discoveries. So the average is about 250 to 300 per year that are documented new disease gene associations per year in OMIM. And so I find this tree a very important concept, because we have the bottom where this is the 30% that can be diagnosed by that test that we now have available. But it's very important not to forget that within that data, there is discovery. And so we don't need to start all over again. We first need to look at what that existing data potentially might tell us before we move on to the more expensive technologies. And then this really focuses around some of our national aims about ensuring we have a mechanism to facilitate secondary use of data for discovery. And so, as I said, and especially now since this test is in the healthcare system in a lot of places across Canada, as either mostly as pilots, but will be there as part of clinical testing. Is now what we're doing is we're siloing data all over again, right? So in the research world, we've been addressing this for a decade, but now as this test moves into the clinical world, each data custodian is siloing off its data, and so we need to find a way that we can leverage that data for trying to find diagnoses for these families. And so this is where our Genomics for ID platform comes in. This is a research platform, not a clinical platform. But the thinking is that some of this data that's generated now in a clinical setting can be freed up with the consent of families and brought into a national platform where it's all put in one place to allow both data sharing as well as matchmaking, which I'll tell you about in a second. And this is the platform that this is built upon is PhenoTips. So it includes phenotypic information from those families as human phenotype ontology codes. And we store all of our data sets within Genomics for ID in standardized formats for genomic type data, but also we have RNA-seq data, epigenetics, metabolomics, all other types of data now, which is not as easily curated, but is present. And Genomics for ID allows us, because the families are consented and this data is coded, allows us to share data with partners in 3 different ways. Open is generally aggregate data or candidate gene type data. Control is data where it's been approved by a data access committee that some set of data can go off and be studied in a very controlled way. And then restricted data where you have trusted individuals coming into your data set to discover, and I'll show you an example of that. But the most easy way to share data is called matchmaking. And so when you think about matchmaking, this is what we sort of envisioned about 7 years ago. And you can sort of see down the side, we have 2-sided, 1-sided, or 0-sided. So 2-sided matchmaking are these 2 little guys with their blue puzzle pieces, where they both have a hypothesis. So 2-sided. And they're bringing this into what we call the matchmaker exchange, which I'm going to show you in a second. 1-sided matchmaking is where only a single person has a hypothesis, but can come into a data set like Genomics for ID and make a specific query and say, does anybody in this data set have a set of parameters X, Y, Z, and show me. And then 0-sided matchmaking is really where you put together huge data sets and allow software and AI to do things in terms of trying to find patients within that set who potentially have evidence that you have a new disease gene association. So, as I said, if all of our candidate genes go out from Genomics for ID into Phenome Central, which is a matchmaking node of the Matchmaker Exchange, and this is also in Toronto at SickKids, and it goes out into the Matchmaker Exchange, which allows us then to connect with 134,000 unsolved patients across the world from 104 countries. And that's February's data that I just got. And within that, within Matchmaker Exchange, we currently have just over 15,000 genes represented, which, as you can imagine, if we have 21,000 genes, most genes are now in there that are flagged as a candidate for a particular set of phenotypes. And last summer, we published a special issue in Human Mutation that sort of talks about the Matchmaker Exchange, which has been ongoing since its launch for 7 years, and now is probably at least directly attributable for of about 700 discoveries. It's probably more. You can imagine when you get into the hundreds, it's hard to count, but we can recognize at least 700 publications that talk about using this network to find additional patients to show that you have evidence for a novel disease gene association. And I just want to show you some of our data, which was led by Matt Osmond from my group, where we had 194 candidate genes from Care4Rare. So these are candidate genes that we identified from reanalysis of existing exome, predominantly exome, but some genome data. And this resulted in 1,500 matches when you put that into the matchmaker exchange. That's a lot of matches. This takes 2 days a week to monitor those matches. But over this 2-year experience, I just want to highlight a couple of things. The first thing, of the 194 genes that went in, only 11 didn't make a match, which means you pretty much match on everything you put in. Okay, and that's not surprising since there's 15,000 genes in there. Doesn't mean they're all real. The second is that because you can sort of reduce these matches to about a core group of about 800-ish so connections. And so because there's some duplication or there's cohorts already forming in there, you can kind of strip that stuff away. But that's still 861 emails you have to write. And so if there is phenotypic information and zygosity information, you can rule them out without having to email anybody. But that only happened about 129 times. And when you send all those emails off, 40% don't answer you. So also frustrating. But in the end, when you put the 194 genes, at the time of this slice of writing of over 2 years, 29 genes became a validated, match-validated novel disease gene discovery and were published as part of our work, which is 15%, which is still very amazing. The challenge here is that most Gene Matcher, which has about 65,000 of the patients in it, does not routinely make phenotype mandatory. So as a result, it's just a gene. And most of the time, then you can't rule it out without actually emailing somebody and having them email you back. In the other big nodes like Phenome Central and Decipher and Seeker, which contain less data, but they contain more data about the case. And as a result, you can rule out half of those matches without having to get in contact with anyone. And so definitely we've said that this is a really important thing going forward. So as I said, 15% discovery and encouraging the community to make sure they include the phenotypic information as well as the zygosity of the variants in the gene will really improve this. But all that said, 7 years later, it's still kicking along, and it's really enabled a lot of discovery. I want to just briefly mention one-sided matchmaking because we have just recently, we've just launched the pilot and finished the pilot and about to launch this a bit more broadly. And that's really where we have somebody like a trusted individual who we know is coming into our database, but has an academic position, is trying to find genes, and they come in and say, do you have any variants in gene X, and show me them at whatever frequency, etc. And with that comes the phenotype, so they can rule things in and out themselves. And we've now set this up called a one-sided matchmaking portal for Genomics 4RD. And we're just in the process now of being able to link that to Genomic Answers for Kids, which is from Kansas City, and being able to then make discoveries within our 2 data sets, going back and forth this way. And then we will open up this so that we can have trusted users come in to the one-sided matchmaking platform and put in queries and then see if there's something in our data, which is a really important strategy because we haven't necessarily flagged all the genes in that data set. And this ensures that all the patients could potentially benefit from discoverability this way. Okay, now I want to get back to the stories. And this is because sometimes the genetic etiology is just not detectable to that technology that you use. So potentially it's not in the coding sequence or not easily detectable by the technology that we use to interrogate the coding sequence. And so this is where this next wave of technologies are coming along, and I include short read there, long read, and transcriptome sequencing being the big top 3, but there are definitely others, and deep sequencing has actually been very helpful for our team, though I won't go into that too much today, but looking for mosaic mutations in dominant conditions is it accounts for quite a bit of the unsolved. So it's one thing when we had exome sequencing, we just kind of knew everybody just goes down the same track. It's easy peasy. But when you think about multi-omics and what kind of approach do you use for which patient, it becomes much more complex. And we've done a lot of thinking about this in terms of resource allocation, because unfortunately we're not independently wealthy and we can't give everybody every single omic approach. And so we got together a couple of years ago with some experts across these technologies and tried to envision how we might make decisions about what would be the best strategy for a particular unsolved family. And we published this at that time. And how we did this was really look at the unsolved cohort from a clinical perspective. And you'll see the numbers across the bottom, which is basically the unsolved Care4Rare cohort that we're going to focus on. So I said we have over 3,000 families, but we basically chunked about 1,000 of these to take them through this process. And it's not quite complete, but at the end, I'll show you our data outcomes. But you can see then, so in the first group, these are then the very specific rare genetic diseases where only 1 or 2 genes causes that. So like cystic fibrosis, neurofibromatosis, and the patient meets clinical criteria, but the test is negative. So you know the mechanism must be in there somewhere, you just can't find it with your technology. The second group is the genetically heterogeneous group, so like neuropathy or ataxia, spasticity, hearing loss, where they've had a panel for their clinical phenotype, but it's negative. So you can imagine there's lots of reasons, including novel gene discovery, that would account for that, as well as mechanisms within those genes that have not been detected. The third group is the unsolved recognizable disorders. There are not a lot of these left where you can sort of make a very clear clinical diagnosis, but there are about 10 of them. And most of these have had all kinds of technologies from many groups, including ours, and they're a challenging group. So those mechanisms will be very interesting, but things like facies, for example, where it's a recognizable thing and everyone thinks it's mosaic, but we just can't sequence the right tissue potentially, would fall into that category. And the last, which is our big category, is the previously undescribed disorder. And these are most of the kids who have had clinical exome or clinical genome-wide sequencing. So they don't have a recognizable disease, but they have a constellation of phenotypic features, which made that the appropriate test, and they're negative from that test. So let's go to Colton. And so he was born in 2010, and I first met him at 11 months of age. And you can see his picture there. He had profound hypotonia and global developmental delay. Very happy little guy. He had 4 of these capillary hemangiomas and some distinctive facial features. And I've basically seen him every 1 to 2 years for the next decade. So very long time. He had to have everything, and this is just a little capture of his diagnostic journey. So saw him in 2010, had the usual investigations you would do for a hypotonic infant. They were negative. He came into our first project called Forge. We identified a candidate gene, and then he got a clinical exome. They identified the same candidate gene. That candidate gene may be responsible for hemangiomas, but it's not responsible for the rest. He was enrolled, and that data was reanalyzed. We got nothing else. Moved on to genome sequencing. And I'll show you what we identified, but we basically, without genome sequencing, we would not have found the genetic mechanism for his condition, which is due to homozygous small deletion in the gene TBCK. And so this is just a screen capture of that. And you can see Colton here is in yellow, his dad's in blue, and his mom is in pink. And what this is really showing, as you can see here in this big set of white, this is Colton's genomic area here. He's got a homozygous deletion. Both of his parents are carriers for that same deletion. It spans about 3 exons. So that would not have been detectable by microarray or exome sequencing. Might be detectable by exome sequencing now, but it's still a small enough deletion that might be tough to call. And so he has a TBCK-related condition, which classically explains his presentation. And it's actually a common enough, but a bit non-specific, right? But common enough that we've seen it several times in Care4Rare over the years. And this is one of our Care4Rare groups from London who published a paper. This gene's been around since 2016. And we have several families now within that got this diagnosis as part of our unsolved pipeline. And this is Colton and his parents, who you can just read that there. But basically having some clarity after 10 years, and now connected to other families who have the same condition through all of the great things that exist out there to connect families, including Facebook, etc. So they're not as alone. Now I just want to move to Austin's story. So he's 16, almost 17 now, and I first met him when he was a couple years of age. And he had he was living not within Ottawa, so it was in a rural area, and the family was having a very difficult time with his illnesses as a youngster. So he had global developmental delay, he had low tone, recurrent infections, and some behavioral challenges due to frustration with his communication. And on his MRI, you can see right here, this is his MRI, he has this little image here, looks like a molar tooth, sort of just got a tooth placed on top here. So molar tooth, which is a pathognomonic finding of Joubert syndrome. So in 2007, we did the clinical testing for Joubert syndrome, but there were no candidate variants identified. In 2013, his younger brother was born and passed away from the same condition. And in 2015, we brought the family into the Care4Rare research pipeline to look at, to reanalyze his data. We found one variant in the gene SEP120, which is known to cause Joubert syndrome, but only one, and it's recessive, so you need to have two. And so he went on to have RNA-seq and genomes to identify that second variant. And I won't go into this in too much detail, but Austin is here. And you can see that this is showing us that he has a very complicated splice difference compared to controls there. And then when you go and look at pulling this out and sequencing it, it is quite a complicated splice change, which retains part of an intron. And you can see in the fibroblast cell line from Austin and the lymphoblast cell line from Austin, he's the effect A and the control is C. You can see that the full length protein for SEP120 is decreased across the board. And so this finally, after 10 years, identified the type of Joubert syndrome he has. And here he is here, Austin. He's done some great PR work for rare disease. He did it on the agreement that I would sign off on his high school volunteer credits. And so he's been a great supporter of rare disease. And these are his brothers and sisters. And then the last example I want to show you is these families, and there's 11 now in total, but here they are. And I grabbed this image from a grant that I wrote back in 2008. So I have been looking for the cause of this spinal cerebellar ataxia, which is very prevalent in the French Canadian population living in Ontario, for like 13, 14 years. And so this is characterized by, and I practice in mostly in neurogenetics. So this is characterized by family members who have adult onset balance issues and poor coordination. They have some eye movement anomalies with slow movements of their eyes and often have double vision. It's slowly progressive over time, clearly autosomal dominant. Their MRI shows atrophy of the cerebellum, and all families are Franco-Ontarian. And so we have worked on these families for over 10 years. Exome sequencing, short read genomes, and RNA-seq has been unrevealing for all of them. And I always suspected they had the same thing, but we didn't really have specific proof of that. Turns out they do. And so this is long read sequencing here. And what you can see here, let me get here we go. So this is one of the individuals here. You see individual 22 here, that was 096. And what you're seeing is the normal repeat, but there's an expansion across within an intron of FGF14 that is expanded in all the affected family members. You can sort of see this expansion piece. And this actually has turned out now to be, so it's an intronic GAA repeat expansion, which has explained all the families I have ever seen in the last 15 years. As well as lots of other French Canadian families from Quebec. And we did this in collaboration with Bernard Bray in Montreal. And if we went back and we pulled all of our families from here, and this is just a gel sort of looking at the repeats. So above 250 repeats, we are seeing affected individuals like that. It's a really easy assay. And of the undiagnosed ataxia families we have in the Care4Rare cohort, almost over 50% of them have their ataxia because of this repeat expansion, which required long-read genomes to be able to see it. And so it's quite possible that many of these other families who have not had long-read sequencing yet also have repeat expansions that have yet to be discovered, and that'll be an exciting thing that we start to do in the next year or so. So the outcomes here are we've taken, of the 3,000 families we have, we took a subset of 1,326. It's an odd number, but that's how it goes, and put them all the way through, or putting them all the way through an unsolved pipeline. And the first thing, as I mentioned to you, the first thing you have to do is go back to their original exome data and reanalyze it. And so when we do that, we get a 32% diagnostic yield. So that's huge. You cannot throw out that data without relooking at it before you do all the fancy omics. And if you break down that 32%, what we're seeing is that 9% get a diagnosis in a known gene, because since their last analysis, that gene has been discovered, and it explains their clinical presentation. 8% of them are gene discoveries that we discovered for the first time and published. 2% of them actually got a diagnosis back in the clinic using a different approach. And 13% of them have a very compelling candidate gene that we currently have out in the Matchmaker Exchange, looking for other families in the world that may have the same thing. So of this, what we've done so far, post this reanalysis, we have 841 families that are still unsolved from that primary reanalysis, and 350 of these have been selected to move forward. Now, the reason it's 350 is that each of these families needs to be completely engaged in this unsolved pipeline because of the types of samples that are required for multiomics. And so they have to come in. Sometimes they need to give blood for RNA, or they need a fibroblast or skin biopsy for a fibroblast cell line, etc. So we picked 350 families who had a very compelling phenotype, really have strong indication based on family history or presentation that it should be monogenic, and we got additional samples from them. And so they have now gone into the types of technologies that I showed you for Austin and Colton and for the ataxia families. And our diagnostic yield so far of the 154 that have come out is 37%. And as I said, deep sequencing is a big win here. And so we are finding lots of mosaic mutations in things like families who have been unsolved, who have NF1 or tuberous sclerosis, for example. Short read genome sequencing, like I showed you for Colton, where they've got a small deletion that was detectable to previous technology, are the ones that fall out easily there. RNA sequencing, like I showed you for Austin, and then the long read genome, like I showed you for the ataxia families, are examples of the types of diagnoses we are able to make. And I should stress, at least at this time in the unsolved pipeline, those other omics are basically finding mechanisms in rare diseases we already knew the genes for. So it's not discovering new ones as much right now as it's discovering mechanisms in the old ones. And I think that's important. I don't think that'll stay like that necessarily forever, but the data analysis around these types of technologies is not minor. And so it's been easier sort of to pick off ones where we know that the phenotype matches what we understand about that particular rare genetic disease. But it'll come. But right now, these are not, with the exception of long read, where you can find novel repeat expansions, but by and large, they're finding mechanisms in disease genes we already knew about. And so what we want to be able to do in the end of all this is find a diagnosis for all. And so, as we mentioned, clinical exome sequencing, something that's whatever it's exome or genome, but it's focused on the coding region, will give you a diagnosis about 30% of the time. And then if you reanalyze that data a couple of years later, you'll get another 5 to 10%. And the novel gene discovery from that existing data is probably max, probably 20%. So you can see the coding genome, probably 50 to 60% will give you answers depending on your inclusion criteria of the families you're studying. But then this is sort of vague over here, and it's just sort of an estimate. But some of these genomic alterations and gene regulation things that we're discovering with short read and long read sequencing, as well as RNA-seq, is helpful. And the mosaicism, when it's targeted to the right set of genes, is very helpful. This group of individuals with suspected rare genetic disease that might actually be responsible, might actually have complex inheritance, probably does exist. And our tools right now to decode that are less than ideal for sure. But it's something that I think making sure we keep this data and make it available for discovery will be very important going forward. And then the last thing I want to just touch on today in this one slide is that there is such thing as a post-diagnostic odyssey. And so diagnostic clarity is great, and the further we can move that ahead in the diagnostic care pathway is the goal. But after that, there's a second odyssey that comes, and that comes from being diagnosed with a rare disease, and it's rare. And so these are the types of challenges that you can see that are the focus of today's events and recognition of rare disease internationally. But the lack of expertise and treatments and access to treatments and the care coordination, particularly when individuals with rare diseases become adults, is challenging for all, and something that we can't not pay attention to. And so with that, I would just like to wrap up so we can have a few minutes for our questions. But I want to just make sure that we recognize all of the contributions of this large network that I'm representing today, as well as all the funders over the past 10 years, which has been a remarkable opportunity to help lots and lots of families achieve their diagnostic clarity. So thank you.
Thank you so much. That was a very, very fascinating presentation. It's good to have this overview of all the achievements we've had over the last little while. You also touched on several challenges throughout the presentation, some more locally here in Ontario and some across Canada. From your perspective, what do you think is the single biggest challenge in mainstreaming this type of patient care model across Canada in a clinical setting?
That's a good question. I think it's resources, probably, and an organizational structure that comes from the top. I think lots of places in Canada and the world who have been able to sort of really focus on undiagnosed families through things like undiagnosed disease clinics, et cetera, et cetera. But all of that requires additional resources that in our healthcare system we just don't necessarily have or can't allocate it to it in that fashion. And all of that needs to also be linked in with a research program on the back end to do the things that are difficult and can't be done in the clinical mainstream. So having something that is really connected is the thinking, but very challenging, obviously, to set up across different jurisdictions.
Yeah, certainly. So I think my next question was going to be sort of around the sort of the research, clinical frameworks and consent frameworks here we have in Canada, sort of compared to a national research project versus what would be a national strategy for sort of minimizing the province by province or territory by territory public healthcare infrastructure. You had a picture of those grain silos. How do we get rid of those grain silos? Is there something that can be done in the short term and the long term? Do we rely solely on research consent and research platforms for that sort of data sharing, or do you see a longer term vision?
Oh, that's a good question. It's one we've been thinking about for several years, because there's a real equity issue here in Canada or elsewhere. It's not just a Canadian thing. And so if you're seen at a center that's got a great research program and you're undiagnosed, you will get access. You're probably going to get access to the research program, right? If you want it. But that's not going to be the same across different places. And so we had lots of discussions within the Canadian community about how to address this, because there's a certain amount of data you can share between clinical institutions, but you can't, unfortunately, share it for discovery per se. As much as we would like to, and as much as we actually personally think that should be part of care for rare disease patients, the ministries of health don't see it that way. Anything that is considered research is outside their purview. And so that's where we came along with this thinking around Connect. And so having a national platform that allows families to say, yes, you can have my data, and yes, you can contact me for research, gets away from you having to have all of these sets of resources across, 25 sites in Canada, and instead centralize it, which we hope will help address the equity issue. I mean, it's the thinking, and that's what we're going to pilot, and then we're going to have to show the utility of taking that approach.
And I guess taking that a step further, like we have a lot of great academic research centers here in Canada, a lot of which are doing cutting edge things in rare diseases or other disorders. How do you see the role of genomic care extending out of these tertiary care centers and pediatric hospitals into other regions of Canada? And how do you think we could do that?
Yeah, so I think that's a lot where we're starting to think about how do we get the patient to the right test in the shortest amount of time. And I think the current model where you, as we showed in the diagnostic odyssey, where you loop around and eventually you might get to a genome-wide sequencing test, but it might take you 2 years, needs to be flipped on its head. And I think the role of specialists who have expertise in genetics probably needs to come not spending our time getting the people to the test at the beginning, but being on the back end of that genomic data and helping interpret that test. And so as we think about a model which I would refer to as mainstreaming, which is the concept that the test would be flagged as potentially appropriate for a patient up front and can be ordered up front, but then the specialist who can help interpret that test sees on the back end would take away most of that work and allow us to focus on finding diagnoses for these families instead of phenotyping them and discussing the test, et cetera, right? You got to flip that triangle basically.
How do you think that applies to either more rural or northern communities, sort of to Austin's story from your presentation?
Yeah, that's a great question. So we're piloting a couple things with that, but one of the things we're piloting right now is something called self-phenotyping. And so as families seen in Ottawa are undergoing genome-wide sequencing as part of our pilot project, before they get their test, the family is phenotyping themselves, and then the clinician is phenotyping them. And what we're doing then is comparing the family reported phenotype with the data to say, can you actually get that diagnosis based on what the family thinks? So it's a kind of a neat way to think about empowering families to get themselves to the test, even if they, live in a rural place in Canada and have a family doctor. We can still support that family doctor through appropriate training, but can the family actually contribute to that process as well? Well, we'll have to wait and see. In our initial pilot, the families did very well, but again, it's a bit biased still.
Yeah. I have another question for you. So as part of, you spoke a little bit of the Genome-Wide Sequencing Ontario project. So as part of that, are we actually sequencing more patients compared to when out-of-country testing process was being funneled through the Ministry of Health? And have the criteria, become easier to order those tests?
The criteria are exactly the same.
Okay.
So that has not changed. That was part of the pilot, because when we were previously in all Canadian places, when they were previously sending things out of country, we had no idea about the outcome. So you're funding a several $1,000 test and the payers don't actually know what the result was. Was it positive, negative, what have you. So the criteria in Ontario were exactly the same as they were previous when we were sending things out of country. I would say there's been an increase in families that have access, but it's not ginormous. It's not tripled or anything like that. It's kind of constant. And I think that what we would do going forward is evaluate changes to that clinical criteria and the outcomes with that to see if we can't broaden the eligibility criteria. Just because a test has a diagnostic yield of 30%, well, that's an arbitrary number. Why does it need to be 30%? Is 10% good? That comes all into the cost effectiveness studies that payers need to see when they decide if they're going to fund a test or not. But just because it's 30 doesn't mean it always needs to be 30. And in which case, can you broaden the inclusion criteria to capture more diagnoses?
So we have some questions that came in through our audience. So one is, are there specific criteria to decide between RNA sequencing, long read sequencing, methylation studies? in the undiagnosed cohort post-exome sequencing and reanalysis?
Yeah, there are. Basically, it depends on what the post-exome reanalysis showed us. So, for example, if the post-exome analysis identified one candidate variant in a recessive gene that really looked like it would be a good match for the patient, but we're missing the second one, they would go to the RNA-seq genome combination. Right away, versus if we had a patient with tuberous sclerosis who had negative testing, they would go to deep sequencing right away. So I didn't show that today, but we do have very painful to look at decision trees on which technology they get and when. And for I think you also asked about the methylation studies. We collaborate with a couple of researchers in Ontario, but Beckham Sideric in London in particular, such that most of our undiagnosed go to EpiSign to see if we can get a clue. And we also use EpiSign for those where we have a variant in a singleton or a duo and can't tell whether or not it's actually de novo to see if the signature matches that particular recognizable signature disease. So I think that the key part of all that is it's very custom, right? Each family is its own unique way of doing things at the moment. Now, that won't necessarily always be the same as the new technologies come along that can do RNA and the epigenome at the same time as your long read, right? Even that exists now. The data analysis for that is horrendous, but it'll get better with time. And maybe there'll actually just be one assay that we can run on everybody.
Yeah, that would be wonderful.
Yeah, it would be. Save a lot of my time.
We have another question from the audience. I'm assuming this is in the context of either Canada or Ontario. Has diagnostic testing expanded to whole genome sequencing?
That's a good question. Diagnostic testing in Ontario for whole genome sequencing is not funded at this time. And then across the country, there are a number of pilots that are using genome sequencing, but they're not actually reimbursed within that healthcare system yet. And so I think we are still developing the evidence to show that genome sequencing is superior. And a lot of what we've done at GSO did not set out to show that it was superior, not by intention, but because one of the criteria for GSO is that you had a microarray. So therefore, all of the things that you would have diagnosed on genomes potentially are gone. And so your genome essentially becomes a coding genome. And so we need to make sure that we can use the genome to its full potential to show the cost effectiveness of that approach. Or the genome cost needs to come down considerably. Once it's equivalent, you can just switch them out, but that's not happened yet. But maybe it will by next year.
So we have a few more minutes. We might not be able to get to all the questions, but I'll try to get a few more out there. One audience member is asking, what do you think the trend would look like for countries, regions such as the Middle East and East Asia that do not currently focus on rare diseases much? How long would it take them to catch up to what is currently happening in Canada, US, and Europe? And do you think with those advancements, it would be faster? And would social stigma play a role?
That's a good question. So I think that a lot of what others have developed could certainly be used, which would save lots of time and infrastructure. It would still take some resource allocation, obviously, to bring those things in, and training programs to make sure everybody feels comfortable with how they're interpreting the data. So I think that it could come faster if the resources were there. How that particular region or country prioritizes the diagnosis of rare disease for their population is very country dependent. And certainly there are lots of developing countries that we've had interactions with through some of the international consortia that I work with, where they have other problems that they're trying to deal with, and rare disease is not top of mind. Doesn't mean that there's not lots of undiagnosed rare disease that are there. And then there are others where social stigma may potentially play a role. And I guess, again, that's something that would have to be addressed very purposefully.
Yeah, and I think relying on the community of people who really are passionate about this, across various countries is actually a really good way of starting up rare disease or undiagnosed research projects in countries that haven't done this. ability or have connections to people who are interested to opening that up. There are great resources like the Undiagnosed Disease Networks International and others that have experience in starting up those sorts of programs in both developing countries and in developed countries and sort of across the board. So that would be a great resource to start off with. I think with that, we have, that's all the time we have for questions. So I'd like to thank Dr. Kym Boycott for being part of today's speaker series. Once we finish the webinar, you'll see a feedback link in your browser and an email for you to fill out just a minute or two to offer some feedback about the webinar. The email also gives you access to all 22 previous episodes. We've done so many already on demand. You can sign up to receive alerts about upcoming speaker series as well. And please stay tuned for our next episode in not too long. Thank you very much, everyone.
Thanks very much. Thanks for having me.
Our pleasure.